Hello everyone, my name is Sabrina Locatelli and I'm a researcher at IRD. I'm based in Laos since August 2021 and I'm going to talk about the Onenakai project, a One Health approach to estimate the prevalence and genetic diversity of gastrointestinal pathogens circulating among elephants, domesticated bovines, and the human population living at the edge of the Nakai Namton National Park in Laos. This project relies on the collaboration between partners based in Laos, from the University of Health Sciences, the National Animal Health Laboratory, and the Conservation NGO, and Malaysian partners from the Wildlife Department in Sabah. This park is one of the identified key biodiversity areas within the Indo-Burma biodiversity hotspot. So since the impoundment of the Nakai reservoirs, elephants have moved close to villages, and there's been an increasing evidence of human-elephant conflicts of the plateau across a wide region. This is leading to an increasing exposure of pathogens for the elephants, domesticated animals, and the human population. Among the pathologies transmissible from elephants to domesticated animals to humans and vice versa, there are gastrointestinal diseases like, for example, soil-transmitted helminths, which are responsible for helminthiasis. These infections occur through ingestion or skin penetration of the infective third stage larvae or eggs present in water, food, or soil, and represent therefore a potential zoonotic risk when human and animals share the same habitat. So helminthiasis are a burden for millions of people across the world, especially where access to water, sanitation, and hygiene is poor. So because in this national park domestic buffaloes and elephants congregate at artificial salt licks, we are expecting transmission and parasite sharing among these animal populations. We are expecting rural human populations living in proximity to be infected with gastrointestinal parasites of zoonotic origin. Our general objective is to conduct a pilot study of gastrointestinal parasites prevalence and genetic diversity on animals and people living in villages frequently visited by elephants around the Nakai Reservoir. Our first objective will be to collect about a total of 200 fecal samples, and we're going to conduct a preliminary prospection and information visit to select villages, and after that we will proceed with the collection of stool samples, so in humans, domesticated bovines, and elephants. Our second objective will be to determine the parasite prevalence and genetic diversity. We will collect fecal samples from families who own domesticated bovines, which congregate with elephants at salt licks hotspots, and store them in different media for parasite detection via microscopic analysis, but also for DNA extraction for further molecular analysis, like, for example, microsatellite analysis to discriminate individuals within the same species, and amplification of parasite target genes fragments. This analysis will take place at the Sabah Wildlife Health, Genetic and Forensic Laboratory in Kota Kinabalu in Malaysia, and sequencing will be outsourced to Peninsular Malaysia. The work schedule is planned as following. So from February to April 2022, we will focus on field preparation and the administrative authorizations requests. At the end of May, we will conduct a prospective tour to select 5 villages, followed by a preliminary information mission and participation signed agreement. And at the beginning of June, we will start the collection of the human stool at the villages and elephant and domesticated cattle fecal samples at 8 salt lick spots. Until October 2022, we're going to conduct the laboratory work and analysis, and the months of November and December 2022 will be dedicated to the scientific valorization. We expect to obtain a first assessment of gastrointestinal parasites circulating among human elephants and domestic bovines, and the domesticated animals and the human population infected will be treated with ad hoc anti-parasite drugs. The phylogenetic comparison of pathogens will shed light on the zoonotic potential of the parasites detected, and the sample collected will be stored for future research on different pathogens, and elephant samples will be integrated in a population genetic study to evaluate their genetic fitness and population size. And of course, students will be trained, and we plan to publish in a parasitology or One Health related peer reviewed journal. I thank you very much for your attention.